Nanoemulgel as a topical carrier for hydrophobic NSAID comparative in vitro diffusion studies. Introduction. The skin represents the most extensive and readily accessible organ within the human body. Pharmaceuticals can be administered topically onto the skin to facilitate either localize or to give the systemic therapeutic effect. Topical administration is a non-invasive alternative that bypasses the first-pass metabolism, reduces the GI side effect, and improves the patient compliance. Topical drug delivery system basically include traditionally ointment, cream, paste, but they causes the stickiness and often causes the patient incompliance. Among them, the gel is become more popular nowadays in both pharmaceuticals as well as cosmeceuticals industry due to their non-greasy nature and ease of application. They are basically formed by incorporating the drug into the hydrophilic colloidal matrix. They are the ideal candidate for the water-soluble drug. Incorporating the hydrophobic drug into an aqueous gel base remains the challenging. Here, the drug X, when given by the oral route, it causes the extensive first pass metabolism. But when the same drug is administered via topical drug delivery system, it overcomes all the disadvantage which is caused by the oral route. When the drug X is converted into the nano emulsion, then the nano emulsion we incorporate in the polymeric gel base, and the nano emulgel get formed. The formed nanoemulgel shows the enhanced absorption as well as viscosity and ease of application for the patient. Objective: to formulate and evaluate the nanoemulsion of BCS class 2 drug, to convert the nanoemulsion into nanoemulgel for topical application, to evaluate the nanoemulgel. Rationally, the drug X is basically the BCS class 2 drug, which is non-steroidal anti-inflammatory drug, and it causes the extensive first-pass metabolism when given by the oral route. Additionally, the chronic oral use often results in GI irritation, ulcer, etc. Then there is an alternative for the drug delivery approach is the traditional topical formulation, which basically avoided the first pass metabolism, which is non-invasive basically, and patient compliances. So the gel is more acceptable because of its non-greasy nature. So by preparing the nano-emul gel, we improve the in vitro drug release of drug X. Solubility studies. The solubility studies of different drugs in oil, surfactant, and co-surfactant were carried out to select the component of nanoemulsion. By using the semi-quantitative method, excess amount of drug was basically added to each vehicle to facilitate the solubilization mixture, and then the solubilization mixture was subjected to the vortex mixture for 10 minutes, followed by the sonication for an hour. From the given bar graph, it is concluded that the methyl salicylate and menthol shows the highest solubility of the drug. Then the twin 80 shows the highest solubility among all the three surfactant. In case of co-surfactant, the propylene glycol shows the highest solubility of all the three. Emulsification studies. The emulsification ability of each surfactant was determined by mixing the adequate proportion of oil is to surfactant. These mixtures were then heated to 50 degrees Celsius using the water bath. After that, the 50 mg of this mixture was weighed and added to 50 ml of volumetric flask. Millipore water was added to that volumetric flask and was thoroughly shaken. Then the resultant mix was allowed to stand for 2 hours, and then percent transmittance was measured using the spectrophotometrically at 650 nanometer. For screening of co-surfactant, the ratio of oil is to surfactant is to co-surfactant is selected. Among oil is to surfactant, the methyl salicylate, menthol, and twin 80 shows the highest transmittance, which is 100% transmittance. Oil is to co-surfactant is to co-surfactant. In this category, methyl salicylate, menthol, twin 80, and propylene glycol shows 99% of transmittance. Pseudo-ternary phase diagram. Here we basically done the pseudo-ternary phase diagram for blank preparation. So the X mix is 1:1. 2:1, and 3:1. Among all three of them, 3:1 shows more nanoemulsion region. So for the drug loaded system, we also select the 3:1 ratio because it shows the more nanoemulsion region as compared to the 1:1 and 2:1. So from 3:1 S mixed pseudo ternary phase diagram, we select the 20-50-30 ratio for preparation of nanoemulsion. Preparation of nanoemulsion. From this 20-50-30 ratio was selected for the preparation of nanoemulsion. The drug was basically dissolved in the oil phase using the vortex mixing to ensure the complete dissolution. Subsequently, the surfactant and co-surfactant were incorporated into the mixture, and which is allowed for some time of standing. Then by addition of appropriate amount of water, the resultant mixture was then vortex to form a nanoemulsion. Preparation of nano-emul gel. The gel base was prepared by incorporating the gelling agent, which is carbopol 980, into the purified water. Then the drug-loaded nano-emulsion was prepared and added to the aqueous gel base, and which is then homogenized for 1 hour to get a smooth homogeneous nano-emul gel. Finally, the pH was adjusted using triethanol amine, and final volume was made up using the purified water. Evaluation of nanoemulsion, particle size and PDI of Ne, that is nanoemulsion. Z average is 183.43, PDI is 0.289. Zeta potential of nanoemulsion is minus 20.2 millivolt. Evaluation of nanoemul gel. The peak 1 shows 264 nanometer. The Z average is basically 189.9 nanometer, and polydispersibility index is 0.283. Evaluation of nanoemul gel. Appearance and pH. White color smooth homogeneous is the appearance of our nanoemul gel formulation, and the pH is 5.12 plus minus 0.080. Drug content 98.56 plus minus 0.89%. Viscosity 135,000 centipoise. Excretability shows the good excretability result and ease of application. Spreadability 6.5 plus minus 0.3 grams centimeter per second. Selection of diffusion medium. In order to show the solubility of drug in various buffer, we done this study for the selection of diffusion medium for the drug solubility. Different diffusion medium is selected, like phosphate buffer 6.8, phosphate buffer pH 7.4. Among all of them, the last one, the phosphate buffer pH 6.8 plus menthol 70:30 ratio shows highest solubility of the drug. So we selected this medium as a diffusion medium for our further studies. In vitro release studies. The in vitro release studies was performed using Teledyne Hansen diffusion cell fitted with the supermembrane, the hydrophilic polyether sulfone, such that one side of the membrane was exposed to the receptor medium and other side remained in contact with the donor compartment. The receptor compartment was then filled with the diffusion medium. The diffusion medium was continuously stirred with a magnet at a constant rate. The temperature around 37 plus minus 0.5 is controlled. The 0.3 gram of sample of each formulation, that is developed nano-emul gel, developed nano-emulsion, conventional gel and marketed nano gel, was placed in the donor compartment, and aliquot of 1 ml was withdrawn at a different time interval, like 0.5 hours, 1, 2, 3, 4, 5, 6, these respectively, and then immediately replaced with the receptor media. The sample were analyzed at 279 nanometer using UV spectrophotometer. Here from the graph, we concluded that the drug release of different formulation for 6 hours is basically the developed nano emulsion shows 99.4% of drug release. followed by the conventional gel shows 33% of drug release. The marketed nanogel shows the highest percent of drug release, which is 100%. Compared to that, our developed nano images shows 83% of drug release. SWOT analysis: strength: enhance the skin penetration and drug absorption because of its nano size. Combine the benefits of both nano emulsion and gel. Weakness: surfactant toxicity risk may be there. Then opportunities: growing demand for more effective topical formulation, expanding the market for patient friendly formulation. Threat: regulatory approval challenges, competition with other advanced drug delivery systems. Conclusion: developed nanoemulsions serve as a promising topical delivery system for hydrophobic drug. NSAID exhibit improved solubility, viscosity, and drug release characteristics. In vitro diffusion result confirm its potential as an effective alternative to the conventional gel. And last but not least, our developed nanoemulsion achieved the drug release comparable with that of marketed nanogel. These are the references, and thank you.